So I'm Ken Oostra. Just give you a little background of myself. I actually grew up on a dairy farm west of Linden. 1986, we actually went on the buyout, so that was the last time. I milked cows, went off to college, they went away. I've been doing lab work for 25 years. I worked for other health systems, ran that lab for 12 years, set up Bellingham office for Edge Analytical, set up their whole microbiology department, then went out on my own. I served on the Delta Water Association board, so when we start talking nitrates, Delta has a long history of nitrate issues, and I've been in more nitrate meetings than I care to be at with DOE, DOH. Was part of the 1997 study. So a lot of the questions you guys had about nitrates, I know them, and if you have some questions, I might be able to answer some of them on my clicker. All right, so one of the things I'm going to do is I brought physical aids. So jelly bellies. I'm going to talk about jelly beans a lot because it's a nice reference. When I was young, jelly beans were colored and they all taste the same. That's pretty much, this is representing fecal coliforms. So a fecal coliform. Then we got into PCR. What jelly beans then became is we added flavors to the colors. And, it gave us more treats, more flavor, so it was good. Then we went into sequencing. Now we're at jelly bellies. Jelly bellies is no longer licorice, orange. We now are root beer, Dr. Pepper, popcorn flavors. They're really more complex. This is DNA sequencing. That's how the difference really visually you'll see in the presentation, because I'm going to go over what we do and how it compares to what's been done. The big thing you guys have to understand is one of the biggest factors in every test run is sampling. When is the sample taken? How is it taken? How much water we're doing? You go out to the stream and you pull a sample, you turn around and take a sample 10 seconds later, it's not the same water. So what happens a lot is everybody goes, hey, they took a sample, I took the sample the next day and it was different. Well, guess what? That water that they took yesterday is down the nooksack somewhere. The water you took came from upstream. It's not really comparable. And when I pull this, if this is my water sample, what's the likelihood of pulling this sample to find a black jelly bean? How much sample do I have to pull out of here to get that black jelly bean, which is my fecal coliform? So the likelihood, you got to take maybe 10 samples to find that fecal coliform. So you have to understand sampling and how it plays into every test that's run, because if you don't understand that, all these results are going to be hard to evaluate. So everything's based on sampling, so you have to understand that. So I own Exact Scientific. We started in molecular division about a year and a half ago. I'm just the guy talking. The brains behind it all is Jenna and Casey. They are the real scientists, and they actually are here, and I want them to stand up. They didn't know this. So these two are the ones that are doing that test, doing the sequencing, getting the samples, filtering, and get them to a point where we can put them on the sequencer. And then we work with Brian. And Ryan is the person who's taking all this data we're generating. And then there'll be a slide and you'll see how much that represents the data. And he's the one that's parsing it through, running it, and analyzing it. Right now, just to give you how much power it takes, we sequenced the Scott Ditch samples, just the fecal coliform parts. It's been 48 hours and we're still running through that data through the computer. So, and that was 16 samples, so that's not even all of them. But there's a lot of information that has to happen there. So let's get into, is this working right? What's the?
Just the arrows. Yep.
Okay. What I'm going to do is I'm going to teach you and educate you on DNA, because terms you need to know on DNA and what DNA means is very important so you understand it. So DNA is the carrier of genetic information in all living organisms. eDNA is DNA extracted from environmental samples, water, soil, air. So that's when you see that term eDNA, that's what it's referring to. A gene is a distinct segment of DNA that is a carrier of the genetic information. So it's what gives you blue eyes, blonde hair. And then a PCR primer. So you'll see primer a lot. That is a small fragment of DNA used to amplify a specific gene in a sample. One of the genes we did is 16S, which is a bacterial gene for surveys. Microbe, which is your bacteria. So you see the term microbe refers to bacteria and other types of organisms. Microbiome. So this is a term you have to understand. It's a community of microbes. So you'll see studies that talk about microbiomes in the gut of a human. So that is a population of bacteria that are living in your intestine. We're doing watershed biomes. We could look at soil biomes, water, sediment biomes. So those are a group of bacteria. So what we do, just to give you an idea of the method we're using, this isn't new. This is being used. The DNA sequencing is used by the federal government. We actually have a contract with the National Park Service, Great Lakes, where we're using eDNA. So they took a water sample, filtered it down, and we're going to look for types of amphibian species in that sample. So they're looking to identify what type of amphibians lived in that water just by using eDNA. The FDA uses it for pathogen outbreaks. Everybody knows Chipotle, so they took it, they sequenced the bacteria that was found there, and so they could link it to who was sick, where it came from. NOAA uses it to identify coral. Same type of thing. They take a water sample, they filter it, we look for the DNA in that sample to identify the coral that's out there. And then universities and hospitals use this method. So this is a method that we made up. It's been used, it's used a lot. All you people that watch CSI and all these other things, it's not that easy. You just don't get to put something in a little hole, push the button that tells you everything about it. It's a process. So DNA analysis is not only one method, and that's things you need to understand is there's PCR, there's qPCR, there's DNA fingerprinting. So DNA fingerprinting, everybody knows about the static, what happened with the DNA there. That was done by DNA fingerprinting. So that's how that was worked. There's a lot more on here. We forgot to put the file chip, which is another way. It's similar to PCR in a way. It's a microarray. I won't get into details. It's kind of boring, but that's another way for DNA analysis. And then there's next generation sequencing, and that is what we're doing using amplicons, non-targeted sequencing, and whole genome sequencing. So what is PCR? PCR is the amplification of a gene or genes using a known primer to target a gene or the genes of interest. So that is, if I want to know if I've got blue eyes, we target blue eyes. So it's organism specific or universal. There's different ways. It gives you a presence-absence result. qPCR is like PCR, except it's using a graph, and you can quantitate in real time, so that as it's amplifying, we can graph it and look for the amplification step. You can use it to be very specific, you can be universal, you can use it as a presence-absence, or you can get a semi-quantitative, which is based on a standard curve where you know certain bacteria at certain times will amplify at 20, 30, 40, 50 cycles. Next generation sequencing. So what you do is you take the PCR and use an amplicon. We amplify a gene set. One gene, multiple genes, target, universal. So we take that and then we go through sequencing. And then we can also do non-targeted sequencing where basically we just take the whole jar, look at every sample within that jar, and just it represents all the organisms. It's not specific to one type. So we'll go over targeted versus non-targeted. And the type of DNA analysis is based on the question we have. So that's the thing. There's all these tools. We have all these tools in the box. So depending on what the question is the tool we're going to use. So it's not like one is better than the other. It's just what's the question. So based on the question, we decide what we want to use. So we use a targeted approach. So we're getting to the jelly belly things. So the question here is, are there red jelly bellies in the sample? So you have your jar of jelly bellies. So we can use PCR or qPCR to answer that question. So we use a primer set, which is that specific gene, and we target one specific red jelly belly. So that's what we're going to look for. So that PCR will amplify to detect the presence of red jelly bellies in the sample. The sequence PCR amplification product, then we take the sequencing of that. So we amplify it and say, yes, we found a red jelly belly. So yes, we found a red jelly belly in the sample. What we do is then we take it through sequencing. So where PCR will say yes, no, it's there, sequencing gives us a lot more data. So not only will we say is that red jelly belly in the sample, but that red jelly belly is also a strawberry flavored jelly belly. So it gives us a lot more information than just PCR. Non-targeted approach is how many varieties of red jelly bellies are in the sample. So we use PCR using universal primers targeting all red jelly bellies present in the sample, including jelly bellies that contain red. So not only are we just looking for the red ones, we're looking for the red orange, the red yellow. Anything that has red in it, we're going to amplify and look at. So then we take it and we sequence those amplified jelly bellies from step 1 and use all the sequences of all red jelly bellies in a sample to differentiate the flavors. So when we do this, we can say that there are 8 jelly bellies that are a variety of red, including closely related colors such as red yellow, red orange. And then these flavors are identified. So when we get into, we say red strawberry, red orange is strawberry orange. So we can get a lot of information. So question 2 is, what colors and flavors of the jelly bellies are in the sample? So we do the whole genome, the whole sample sequencing approach with no PCR step involved. So we're not amplifying anyway. So we're doing a non-targeted, taking this here, sequencing for everything in this jar. So what we can say then is there's 27 different colors and varieties of it. Each color corresponds to an identified flavor. So blue with blue spots are blueberry pie. So we get that specific. You can go into shapes, sizes, different things with sequencing because there's a lot more data available than PCR. So PCR versus sequencing. Targeted PCR requires knowledge of what to look for. So I need to know is, you want to know that there's red jelly bellies in the sample. So then I go looking for red jelly bellies. A lot of times PCR is a preliminary result, and you need a confirmation to verify or validate that result. So also with PCR you can have some issues. So you may say, is, well, I wanted red jelly bellies that are only strawberry. Well, we only can tell you red jelly bellies, so we may find the cinnamon ones as well, which is not what you wanted, so you get this false positive result. The benefits of PCR is it's less effort than sequencing, and it's more cost effective. It gives you a presence absence and can be semi-quantitative. Whole sequencing, on the other hand, gives us more information. So we can say is jelly belly equals red, very cherry flavor, jelly belly red, cinnamon flavor, jelly belly yellow and white, buttered popcorn flavor. So we're giving you, not only are we looking for colors, we're also telling you the flavors, where PCR will only tell you the colors, but it takes more effort with more costs. You need a database with references of colors and flavors are needed because you are taking that sequence and comparing it to a known. But we have so much data, there could be many different questions can be answered with all the large amount of data that's produced by sequencing, where PCR is very specific. Sequencing is very broad. So, which gets us to Scott Ditch. So, Scott Ditch is phase 1 of a study. So, we're going to use eDNA sequencing to evaluate the microbial community of Scott Ditch. We're going to look at input source profiles and monitoring sites. So, what we're trying to look at is the microbial community of the inputs, such as the wildlife refugees on there, the berry farm runoff, the housing development. Are they different, or do they all look the same? And then is there a unique identifier in those inputs that we can use to track to the watershed? So multiple methods we're using to do this study. So the whole sample sequence of membrane filtration fecal coliforms. So we're still running fecal coliforms on that sample at the same time we're pulling our water for sequencing. So what we're doing is taking those fecal coliform results and we're sequencing the isolated fecal coliforms to see if we have different strains or different varieties show up. Are those strains unique to an input? Are they universal strains? Seeing if we can use known testing now, and can we take that known test that we've been running forever and run sequencing on it and get that information. Then we're also using a universal primer set sequencing and looking to see what all the organisms are in the sample, where then we can start comparing those samples using those universal primer set. So here's the current method. So fecal coliform analysis. So fecal coliform is an indicator test. It's a multiple tube or membrane filtrations. To do this test, we have to dilute the sample. So we take a 100 ml sample and we put it through a membrane, and we may put 50 mils or 10 mils or 1 mil. So when you see a count of 300, we didn't count 300 bacteria. We may have counted, if we use a 10 mil sample, we counted 30 of those bacteria. So there's all multipliers. That's why when you look at coliform bacteria, it works at a log 10 plus or minus. So that means 10 and 100 are the same number in the world of microbiology. It's also used to indicate contamination. So what we're doing is we're taking this fecal coliform analysis, which says there's our fecal coliform, boom. Now we're saying is we're going to sequence it. We're going to tell you, is it E. coli? Was it Klebsiella? Was it Citrobacter? Was it E. coli 0157? Was it E. coli 0126? So sequencing is going to give you all this other information, and with that other information, can we pinpoint a source? Here's what the EPA study. This is what the EP study, in a basic term, did. So to the far right is the number of organisms it looked at. So it's color coordinated. So column 9 has what they looked at. 1 through 8 are the samples. Each represented different, we found 3 of these, which could have been the ruminant, could have been the avian. Those colors are organisms identified, so that's the EPA. That's what we did. So each color represents a bacteria. This is an actual set of samples from WCA. We did this past fall. So when you compare those to this, compared to what EPA did, compared to what fecal coliform did, are very different. So this is when we pull the Scott dish samples and start doing our sequencing, this is the data we're generating. So to really make it clear, that's what's run now. This is the routine stuff. This is actually more than what's happening now. This is after sequencing that sample, that fecal coliform. This is what EPA did. This is what we're going to do. Just make sure it all sinks in. That is the difference in what we're looking at. What's next? We need a reference database. So what's happened is people have built some references to different sources, but they've done specific. Get back to that EPA file. We looked at one thing, did eight stuff. So that's all we can look at in a source. I need it to look like that. So we need to run a reference database using whole genomic sequencing on these type of references, so that when we do go in a watershed and we do pull a sample, we have something to compare to. Case in point, jelly bellies. I cannot identify jelly belly flavor based on looks, but I don't have the reference table to look at. So if you grab the yellow white, is it popcorn or is it caramel? We don't know until we look at the chart. We don't know going into a watershed and pulling a sample what's going to be there without referencing something. So a database that will look at urban runoff, that will look at dairy lagoons, so we can compare the samples is needed. and hasn't been done that we know of. We don't know what we don't know. We're going to generate massive amounts of data. Like I said, we're doing a fecal coliform only sequencing right now. It's taking 2 days, and it still wasn't done. Now to generate that big graph is going to take days, and we're going to take so much data we don't know what we don't know until we look, and that data lasts forever. So questions that we thought about asking may be answered in the future, and we can go back to that data. So it's also comprehensive data. That means there's a lot. We're not looking for one thing, we're looking for everything. So years from now, we might be able to go back and say, hey, we have this data back then, and I discovered something now. Was it then there back then? So we can go back in history and find it. And we're not relying on historical indicators. We may find out by doing this, there's unique identifiers, indicators in each source that we can go in the watershed and look for. Then we can get that targeted approach for the right things. Fecal coliform is only an indicator. It doesn't pinpoint anything. But by doing the study, we might find something that will give us the information we need. So, questions? Did I muddy the waters? Did it get clear at all?
Wonderful presentation. I really enjoyed that, Kent. Question on the EPA study. Based on your understanding of DNA sampling, what's the relative value of that EPA study for us, for information?
The value. Trying to be diplomatic about this now. It gave information, and the information that the most value I got out of it is that approach did not work. It didn't. To give you an indicator, there are at least 4 wastewater treatment plants on the Nooksack. How many samples were positive for a human? 2? Maybe? So that alone tells me is they either didn't pull enough sample, the primer sets they were using weren't specific enough, or was below the detectable limit. So to let you know, is we did some preliminary stuff for sequencing, and fecal coliforms are citrobacters, Klebsiella, and E. coli. When we did our sequencing on our primary stuff, we never saw those. So again, it's the question that was asked and what he wanted to look for is what it tells me is there needs to be more studies, more specific things to look at a large body of water using one criteria isn't the answer anymore. And we have tools that says we can do better. Any other questions? The studies we're doing now are on current water samples. So we went out and sampled Scott Ditch. We were hoping to pull another set of samples, but we had a little flood. And we decided not to sample during the flood time because it would be a point that is abnormal. We've been monitoring fecal coliform counts in the ditch to say is when we see something high, that's when we went out and sampled. We'd like to do one more sampling out there, so we're probably going to want to wait till spring to see what that is, so we don't have a data point we feel is not representative.
What is your hope for next steps?
Next steps, what I'd like to do is, there is somebody sitting out there, to start this database, reference database, because that's going to be a year-long study, and we need to do composites. We have this whole plan. We've sent that to the Conservation District Commission. So we'd like to at least over a year start building this reference database. And in the meantime, you don't have to wait to sample. We don't need a database to start sampling. We can start sampling now have that data and start comparing later. We also, at the same time as we're building that reference database, we want to take fecal coliform samples from them references, run them like we do now, see if there's any differences in those sources using that method. Maybe we find out is that there are specific fecal coliforms that are specific to those sources, and we can then say, the stuff you've been doing now, we have at least another way to check it to see if we can match the sources. So those are the next steps that we see.
So let's just say you have a farm that's having major issues with starlings, okay, and they're worried, there's some sort of contamination coming from somewhere they're not quite sure. What could you do now to determine, could this contamination possibly be coming from a bird source?
Then we go into the sampling is the big thing. So if you've got starlings and say they're all sitting on top of the barn, and you got a drain pipe. So we can sample that drain pipe, look for the indicators in there, sequence that, go into the watershed, see if we see those indicators coming throughout the watershed. So we could also at the same time, again, because we don't know what we don't know, let's do a fecal coliform on that drain spout, see if we go out, if there's different strains of that. There's so many different ways to go here. And then go out into the watershed and see if we're seeing the same strain show up there. We may find it unique by sequencing that runoff from that building. We may say, oh, here's 2 or 3 unique bacteria that are coming from there that we know are starling based, so we're going to go and sample out there. So sampling so that we get representatives from that starling where they're contaminating needs to happen. So we need that source that says this is from the starlings, this isn't from the farm. And then go into the watershed and compare.
So you talked a little bit about the absence or presence of certain DNA. What about percentage-wise? If you have 100% of the bacteria, can you tell what percentage came from ruminants? What came from people? Can you base that out on a percentage in a sample?
Not really. Again, that's how we know, until you do some sources, we may get some ratios, but it's not really going to be hard fact percentages to be able to determine it's 50% human, it's 50% ruminant, and those type of things. Maybe there's an indicator organism we find that we can do that with. Again, it's not been done, so it's a hard question to answer without doing some of the legwork.
So currently, just the testing that's being done is really just absence or presence of certain.
For PCR, yes. For sequencing, yes. And the trouble is with the sequencing, there's so much, like you saw that chart. It's so much different organisms in there. We may find a profile that can give us more information. It's not one organism. It maybe is, we see these 10, and we see these 10, and we see these 10s, and we're saying, okay, this profile is a dairy farm, this profile is a wastewater treatment plant, where it's not so much as a one-to-one relationship, where this bacteria is only on dairy farms. This bacteria is only, we might find that as well. But
So would that be more like your Jelly Belly example? Once you know what everything in the total population is, in some ways now I can go back and count and say I had 14 caramels, and I had 330 total. I had 14 caramels. I had 9 blueberry pie.
it's possible using the data to develop a method that possibly could do that. You get into the qPCR. So if we had caramels, and we say, okay, I want to know how many caramels are in there, we could go back to qPCR and qPCR those, build a graph and say, okay, based on this, we know we're seeing this amount of caramels in there.
I suppose it's still always just relative to your sample.
Right. Right, and it's how you treat it and how you do it. There's different, what we found is we went out and took water samples, and we had to go to a 16S primer, universal primer set, because there wasn't a lot of DNA in the water. We took 1 liter samples, we took 4 liter samples, we took 10 liter samples. The WSDA study was based on 3 liters of water, and there's just not that much DNA. It was so low that we have to amplify it to be able to determine. And once you amplify, quantitation becomes an issue. Any other questions?
So it's too soon to tell because you just began this, but if the profiles, say you said the profile of the farm and the herd and if the profiles didn't vary enough, it could all be inconclusive.
Worst case scenario, we come up and we say, we've done all this work, and you can't tell the difference. You cannot point a finger at farmers because you can't tell the difference between a farm, wastewater treatment plant, urban runoff, because they're all the same. That's the worst case scenario. So worst case scenario says, you can say all you want, but as a farm, we know you can't tell the difference.
But you can already identify human feces, right, with DNA in that. Like, say the CSI, they can pin a crime on someone. I think they're based on their DNA in that fecal feces.
It's different. So you get into that DNA fingerprint. I asked that question yesterday to the brains, and they explained it to me. Everybody has the same DNA, but within the known DNA, there's sections that are variable, and so it's a fingerprint. And then basically I'm looking at a group of sequences, and then looking at that group, these variables that are junk DNA are unique to everybody. So I'm looking, it's a broad picture type thing, and the probability is 1 in 11 trillion that you're not this person based on a profile of all these. This would get into profiles of this genetic code. So it's a whole different way of looking at it when we talk CSI, where we're coming through and we're looking at sequences that are very specific, that are going to identify something. So it's a different way. Can they go and do human? Yeah, if you amplify and just look for that, you can. But if I go out and amplify and I know I target one thing, I can target and probably pull out human, bovine, horse, because that's what I'm looking for, and I'm amplifying to get it. So targeting will say yes, it's there or not there. Looks like it's all got it. It's got farm DNA in it, it's got bird DNA in it, it's got this. So here's a good example. Skagit County did a study, sent the DNA study. They're probably doing the bacteroides type study that EPA did, doing the targeted approach. They sent it to Oregon State University, and they sent dog feces to Oregon State. Came back, dog and cow, and they said it's worthless. Every dairy farmer knows what a dog does. And a dog goes out and eats treats out in the middle of the field, and of course will have cow DNA in its feces because it ate cow manure. So it's a known thing. What happens during the fall? You see geese, ducks, what are they doing? They're out in the fields. What are they eating? The corn. Where are they getting the corn from? Cow manure that passed through. So when you sample a goose or a duck, you'll find cow DNA. So problem is dairy farmers know that, but does the guy in the universities that's doing the study know that? Because when this guy started talking to me, and I said, well, that makes sense. He goes, what do you mean? So anybody that's seen a dog on a farm knows what a dog does. And he goes, well, nobody's ever said that. So part of it is information, and if they don't have the information, how do you make the determination? Me, the basic thing, because I've seen dogs do that. So There's other applications. I have an extra slide if you're interested, but I know we're short. It's a short,
Sure.
just to give you an idea. Hirst Decision. In the 90s, EPA, and I ran this method, used a groundwater under direct influence of surface water. Did a big study. Every public system within 200 feet, that a well within 200 feet of a surface water, had to run an MPA. So we looked for algae, amoeba, cryptosporidium, which already identified that to determine if you were influenced by the surface water. Been done for years, still run the test, still have the ability to run the test. We now have something that's better. We can take whole genomic sequencing, filter it, run that sample, do a comparison to the surface water. So one of the things you could do is use this technology to say, am I under the influence?